All right, so we're going to take a look at cellular respiration and how that affects pH and how that affects your breathing. All right, so here we have a cell and we're doing cellular respiration, right? So cellular respiration is when you take glucose, C6H12O6, and oxygen, and you produce CO2, H2O, and ATP. So the only reason we do cellular respiration is to produce ATP. So you can think of glucose as a $10 bill and ATP as quarters. So if you're at an arcade and you have a $10 bill, you have to make change first before you start playing the games, okay? And the easiest way to make change is take that 10 and you split it into 2 5s, and then you take your 5s and you get change for, you get 5 singles and then 4 quarters for $1. Because really, nobody walks around with $10 of quarters unless you're in the arcade, person, okay? But the average person doesn't have 40 quarters, okay? So here's the cell. And we bring glucose into the cell. That's the first step. You can only do that if you have insulin. So insulin is a protein that helps bring glucose into the cell. And once glucose is into the cell, then we're going to do our next step, and that's called glycolysis. Glycolysis, to lyse something, is to split. So we're going to split that glucose into half. And that does not require any oxygen, by the way. That is what we call anaerobic. No oxygen required for that. So we're going to split the glucose into 2 pyruvic acids, okay? And then those pyruvic acids are then going to interact with oxygen. So we're going to bring oxygen into the cell too via simple diffusion. So the oxygen and the pyruvic acids end up in the mitochondria, and this is where all the magic happens. No, it's not magic. It's science, okay? So this is where all the science happens. Remember Krebs cycle and electron transport chain, or the citric acid cycle and electron transport chain? Yeah, neither do I. So anyhow, all that sciencey stuff happens in there, and in the end result, the products are CO2, H2O, and ATP. Boom! Okay, that's awesome if we have oxygen. If we don't have enough oxygen, we can't do this portion here. So we're kind of stuck with 2 pyruvic acids, and then we ferment those and we produce what's called lactic acid. Now, lactic acid builds up, and we only get 2 ATP out of that. So instead of getting 38 ATP, instead of getting 38 quarters for your $10 bill, you're only getting 2 quarters. But if you're really desperate in that arcade, you give somebody a $10 bill, they're going to give you 2 quarters, you can play 1 game. If you're really desperate, I don't know why you would be, but if you are, it'll work, okay? So you get 2 ATP, but the problem is lactic acid builds up. Not so good. But fortunately, your body can actually run this reaction in reverse, because if we can get the lactic acid to the liver, and if you can get some extra oxygen on board by breathing heavily for a couple minutes after you cross the finish line, we can then cool down, and the lactic acid gets converted back to glucose in the liver. I mean, that's pretty cool. Okay, so either way, we're either doing aerobic with oxygen or anaerobic without oxygen. So let's go with aerobic first. Okay, so we're doing normal cellular respiration. We're producing CO2 and water. Well, the CO2 and the water, they combine to form H2CO3, which is carbonic acid, which then splits to form HCO3-, which is bicarbonate, and H pluses, okay? Conservation of matter, right? 2 H's here, 2 H's here, 1 C here, 1 C there, 3 oxygens, a plus and a minus, or a minus and a plus balance out. So anyhow, the more CO2 we produce, so the more cellular respiration we do, we're going to produce more CO2. The more CO2 we produce, the more H pluses we produce. Well, H pluses aren't so good, because the more H pluses we produce, there's a thing called the pH scale. The more H pluses we produce, the closer to 0 we get. 0s are pure acids, 14s are pure bases, 7 is neutral right in the middle. So pure acids have H pluses. Pure bases have OH minuses. If we take H plus and OH minus, combine them together, we get water, which is why a pH of 7 is neutral. The pH of water is 7, which is neutral. Okay? Now, the pH of arterial blood is 7.35 to 7.45, and pH is one of those scales where it's one of the rare scales where there are no units, okay? So here's the thing. The more cellular respiration we do, the more ATP we produce, which is good, but the more the more CO2 we produce as well, which is bad because H pluses build up, which means we're going closer to zero. Our blood is becoming acidic, okay? Now, we can solve the problem, though, if we can get rid of that carbon dioxide. I wonder how we can get rid of carbon dioxide. Well, let's learn. So check it out. If our pH starts to drop, okay? So we're doing lots of cellular respiration, CO2 is building up, H pluses are building up, our pH is going closer to zero. It will never hit zero because of this very, very tight range. But we're dropping. So our pH is dropping. Chemoreceptors sense that. They send afferent sensory impulses to the control center. The control center integrates it and says, yo, our pH is dropping. We have to fix that. So the control center sends efferent motor impulses to the effector. And in this case, the effector is the diaphragm. And the diaphragm will then contract and relax at a higher rate. We're going to breathe faster. So if we increase our respiratory rate, we're going to get rid of CO2 faster. And if we get rid of CO2 faster, this will drop, which causes this to drop. And if the H pluses drop, we're driving the reaction back this way, and we are actually raising our pH back to normal. The opposite happens if our pH is too high. An example of too high pH is if someone is very anxious and they're breathing rapidly, you're very, very upset, and you're hyperventilating. Okay, it happens. So when you hyperventilate, you're breathing very quickly. When you're breathing very quickly, you're actually getting rid of too much carbon dioxide, which is kind of weird, but you're getting rid of too much carbon dioxide, which means this is too low, which means this is too low. If these guys are too low, that means these guys are too high. So we're driving a reaction this way. Chemoreceptors sense it. Control center says, yo, our pH is actually too high. We need to bring it down. So the control center says, yo, lower our respiratory rate, breathe a little slower. And when you do that, you're going to bring our pH back down to the normal range of 7.35 to 7.45. So why is pH so important? For a couple reasons. Number one is our pH tells us how quickly we breathe, whether we breathe slowly or quickly. Secondly, if we don't control our pH well, we will change the structure of our enzymes, of our proteins, of our channels, of our pumps. So remember way back from bio, if you change the pH of something, you actually change the structure of a protein. You'll permanently unravel it, and it's called denaturing. You don't want to denature your proteins because basically you're tenderizing yourself from the inside out. Think about it. If you have a piece of chicken or a piece of meat that you want to marinate overnight, most of the marinades that we use are acidic. Why are they acidic? Because acids and bases will denature proteins, will change the structure of the protein. You're actually tenderizing your food. Well, if we don't control our pH, you're tenderizing yourself from the inside, which is not recommended. Okay? So that's how cellular respiration and pH and our respiratory drive all come into play. And lastly, we confirmed that the reason that we breathe is because of our CO2 and not oxygen. Because if you put an oxygen sensor, a pulse ox in your finger and you hold your breath, you'll be able to maintain a good pulse ox for the entire time that you can hold your breath, for a minute or so. Or if you're really good, a minute and a half holding your breath, your pulse ox will not change. So we do not breathe because we're low on oxygen. We breathe because we're high on CO2. Thanks.